Namaskar. Today we are going to discuss about the chemical structure of DNA. What is actually the chemical structure of DNA? Here, if we carefully notice that DNA, a DNA basic structure of DNA is here is a helical structure of one is 5 prime and it's 3 prime, another is 3 prime and 5 prime. So what are the different chemical component present in a DNA? In a DNA, there is nothing, but there is a sugar, phosphate, sugar, phosphate, sugar, phosphate. One backbone is made up of sugar, phosphate, in a 5 prime, 3 prime direction. What is actually the 5 prime and 3 prime will discuss later. And another is again of sugar and phosphate, sugar, phosphate. And along with the sugar, purine and pyrimidine bases are attached. That is, nitrogenous bases are present and they form a hydrogen bond between them. So, what are the three major components? One, first of all, is sugar. Second one is phosphate, and third one is purine and pyrimidine bases. What are the different kinds of purine and pyrimidine bases? Here the purine bases are adenine and guanine, and pyrimidine bases in thymine and cytosine. Now, in a DNA, total amount of, according to the Chargaff'S rule, in DNA, what are actually the number of adenine, guanine, thymine and cytosine? It's called Chargaff'S rule. According to Chargaff'S rule, total amount of purine base, adenine, guanine divided by total amount of pyrimidine bases will always be 1. That is equal number. What actually the number of purine bases present in DNA will be equal to the number of pyrimidine bases. Now we'll see that what is actually the structure now. What are the different bonds present in DNA? Now first, sugar is present, and along with the sugar, one phosphate, inorganic phosphate molecule is present. Inorganic phosphate molecule, that is H3PO4. Later it has been discussed here. However, sugar and phosphate, this sugar and phosphate, the bond present between two sugar and phosphate is called ester linkage. This is actually the phosphodiester linkage. Two ester linkage. Sugar molecules will attach to phosphate with a two ester linkage, so it is called phosphodiester linkage. Phosphodiester linkage. Phosphodiester linkage. This is the first bond, that is the phosphodiester linkage present between sugar and phosphate. Second bond is present between sugar molecule and nitrogenous base. Sugar molecule and nitrogenous base, that is guanine, is the nitrogenous base. It is called glycosidic bond. Second bond is glycosidic bond. First bond phosphodiester, and third bond is hydrogen bond present between two nitrogenous bases. So three kinds of bases are there. One is phosphodiester linkage, that is present between sugar and phosphate. Another is glycosidic bond present between sugar and nitrogenous base, sugar molecule and nitrogenous base. Sugar, that is, in that case, which kind of sugar is present, I will discuss later. And third one is hydrogen bond present between two nitrogenous bases. Now, in case of adenine and thymine, hydrogen bonds are 2. Here the 2, and in case of guanine and cytosine, hydrogen bases are 3. However, now I will discuss in details what is actually the sugar. In case of DNA, DNA's full form is deoxyribonucleic acid. Deoxy. D means that is oxygen is not present. Oxygen is separated. So in that case, this is the pentose sugar, and it's 2. It's 2 carbon position. Here's the 5 carbon position. 1, 2, 3, 4, and 5. Its 2 carbon position is devoid of oxygen. It contains only hydrogen. In case of RNA, in that position, one oxygen molecule is present. in case of RNA. But in case of DNA, only hydrogen molecule is present in case of 2 number carbon of deoxyribose sugar. So why it is called deoxyribose sugar? And its one carbon position is attached with its one nitrogenous base by means of glycosidic. As I mentioned here, that is the sugar molecule and nitrogenous base, there is a glycosidic linkage. So this linkage is glycosidic linkage 1, and 2 number carbon is present with a 2 hydrogen atom, and 3 number carbon is attached with one OH molecule. This is a very vital, very interesting thing, is that this OH molecule is the main key OH molecule to which it is attached with phosphate molecule. That is one ester linkage is forming here, one ester linkage with phosphate and sugar by removing one water molecule. Here one OH molecule of sugar and one hydrogen molecule of phosphate together forming H2O. This H2O molecule is released and one ester linkage is forming. So, and in 4 number carbon, it is attached with one CH2OH group and one hydrogen. So why? Its 5' end of this sugar, 5' end of this sugar is free. So why the term 5' end is given, that is the 5' end. 5' end due to its the 5 carbon position of that particular sugar of this backbone is free. So why it is called 5'. Okay. Now this 1 carbon position is attached with a purine base. When it is attached with purine base, it will attach with purine or pyrimidine base. Whatever may be, sugar will either attached with purine or pyrimidine. In that case, sugar is attached with a purine, adenine. In this case, again it's purine, and in this case again it's a pyrimidine base. Whatever may be, in all cases, phosphate is again attached with next sugar, its lower sugar, which is 5 carbon position. That is, phosphate in one hand it's attached with 3' end, 3 carbon position of next upper sugar, and lower sugar's 5'. That is the 5 carbon position. 5 carbon with the 5 carbon, that is the OH group is again attached with this hydrogen bond and forming the water molecule. This is another ester linkage. So 2 ester linkage. First of all, this one, and second one is this one. So 2 H2O molecule is released during the formation of phosphodiester linkage. So one sugar molecule, one phosphate molecule, and one nitrogen base. These 3 together constitute one nucleotide. And when phosphate is not present, only sugar molecule, and that is the deoxyribose sugar, and nitrogenous base when present is called nucleoside. This is the basic difference between nucleotide and nucleoside. In case of RNA, only in 2 number carbon position of that sugar contain oxygen. This is the basic difference. And in case of RNA, thymine is not present. Only in case of thymine, it is uracil will be there. All is same. Only thymine is replaced by uracil in case of RNA. So I am here. I will clear again that one I am discussing about the single strand. This single strand is like that, and this strand is like that. DNA is anti-parallel. That is, sugar molecule is present in that way, and in inverted way in that case. So why its 5' end is open here and 3' end is here, and vice versa. So what happened in that case? The same thing occurred. Is 1 number carbon is here. 1 number carbon is here. 2, 3, 4, and 5, and phosphate bond is present. One most important thing is that why DNA is called acid, because phosphate molecule has free OH bond, OH atom. This OH red, this OH molecule, the OH hydrogen, this hydrogen is free. OH molecule release hydrogen in the solution. In all cases, this phosphate molecule release one H plus ion, We know that is the definition of acid is the substance which release H plus ion into the solution will call acid. In that case, DNA release H plus ion in the solution. So why DNA is acidic? Where is the H plus ion? It is the phosphoric acid's H plus ion. The OH molecules H plus ion is released in the solution. So why DNA is negatively charged also? Its one is negatively charged and it's acid. However, here, what is the Chargaff rule? Chargaff rule is nothing. That is according to the Chargaff, that is the total amount of purine, that is adenine and guanine, and total amount of pyrimidine will be the same. What is the basic example here? Is that, sorry. Here, if a single strand of double-stranded DNA contains 30% of guanine, then what will be the percentage of other bases in the DNA? Its guanine is 30%, then cytosine will also be 30%, and if both contribution is 60%, then adenine will be the 20% and thymine will be the 20%, because the rest 40% will be contributed by two, that is the adenine and thymine. So adenine and thymine AT will be equal 20 and 20%. So why AG, here the adenine 20%, guanine 30% is 50%, and cytosine and thymine will be the 50%. So equal contribution, that is the adenine and guanine, purine and pyrimidine will be the 50%. So this is the basic example. And this kind of questions students will face in the competitive exams. And another important thing is that there the what are the actual position, that is the carbon atom of the deoxyribose sugars, one number carbon atom is attached with what number of nitrogen of the nitrogenous base? The answer is, in case of purine, when purine is attached with the sugar, it will the sugars 1 number carbon and purine bases 9 number nitrogen will be attached. And when when pyrimidine will be attached, that is the thymine, then 1 number is attached with 1 number of 1 1. That is in case of pyrimidine 1 1 linkage, glycosidic linkage. In case of purine, when attached with the sugar, that is the 1 9 glycosidic linkage will be